Shall I start?
That's right. Good afternoon, everybody. Welcome to the IBH webinar for this afternoon. I'm just going to let you know, if you haven't seen the notice already, that this meeting will be recorded. So if you don't want to be seen or heard from, make sure your camera and audio is off. I will hand over to Yan Ching to introduce our speaker for today. Enjoy the meeting.
Thank you, Dan. Hi, everyone, and thanks for joining the IBH webinar. I'm Yunqing from the James Hutton-Institute, and today it's my pleasure to introduce our speaker, Dr. Katie Jeal, from Sainsbury Laboratory in Cambridge University. And Dr. Jeal is a postdoctoral researcher in Madeline Bartlett's group at the Sainsbury Laboratory, and she holds a master's degree in biology and a PhD degree in plant sciences from the University of Bristol. And her research focuses on investigating the genetic and developmental mechanisms driving lemma and morphological diversity in the grasses. And her research combine developmental imaging, transcriptomics, and genetic approaches in comparative models such as barley, blackgrass, and brachypodium. Today, she will talk about her work on the barley mutant leafy lemma 1 and morphological innovation in the grasses. And can welcome Dr. Jeal, and the floor is yours.
Cool. Thank you for the introduction, and thanks to the organizers. So today I'll be talking about morphological innovation in the grasses. So evolution continually reworks conserved developmental programs to generate morphological diversity. And perhaps one of the best known examples of this is in the tetrapod limb, in which the conserved developmental program results in the generation of a bat wing, whale fin, and human arm, all of which look very different from each other and serve very different functions. And we see this principle throughout evolution, with the same core developmental programs being reused, modified, and repositioned to produce new forms. We also see a similar phenomenon in plants. Here we see modifications of the leaf developmental program, and this results in a wide range of leaf morphologies. So we have some simple leaves, some compound leaves, lobed leaves, and entire leaves. And in the grasses, such as barley, we see the leaves having two distinct domains: the sheath, which protects and surrounds the stem, and the blade, which is angled away from the stem for photosynthesis. Evolution also pushes this conserved leaf developmental program towards novel functions. And for example, the carnivorous trap of pitcher plants, climbing tendrils in cucumber, protective spines in cacti, and the colourful bracts of hydrangea and anthurium, which attract pollinators, all demonstrate how the same underlying leaf developmental program can produce structures with vastly different forms and functions. This idea is not a new one. 200 years ago, the philosopher Goethe proposed that alles ist Blatt, everything is leaf, and he suggested one fundamental unit, the leaf, acts as the ground state from which all above ground lateral organs are formed. Today, we have strong molecular support for this with the ABC model of floral development. And here we see that loss of function of A, B, and C genes results in floral organs reversing to a ground state, producing a foliage leaf. And this provides molecular proof of Goeth's theory and evidence that many lateral organs are homologous to leaves. So the modified leaf that our lab is interested in is the grass lemma. And the lemma is a protective leaf-derived bract that encapsulates the grass flower and comprises two main parts: the lemma body, which encloses the reproductive organs, and the awn, which is an elongation from the lemma body. And the lemma functions as a key player in grass seedling dispersal. It provides environmental sensing and also aids in seedling establishment across diverse ecosystems. And the evolution of the lemma and its role in aiding with these functions has been widely considered a key innovation that helped that encouraged the global ecological success of the grass family. Across the grasses, lemma morphology is highly diverse, and we see lemmas that lack awns, awns that are initiating from the distal tip, awns that initiate more proximally, and taking this further from just where the awn initiates, the morphology of the awn itself is also highly diverse. So we have straight awns, twisted genicular awns, and some more unique awns such as this feathery one as well. But when we look at this wide range of morphological diversity, and given that is a leaf-derived bract, it's striking that all of these grasses have the exact same consistent grass morphology with a sheath and then a blade, which is very consistent and uniform, with virtually no deviation. And this kind of brings us to the questions that we're interested in our lab. So what are the differences and the mechanisms that are driving this lemma morphological diversity, given how consistent these leaves are? So how can two organs with the same developmental program show such differences in morphological diversity? And to address this, previous work in our lab by PhD student Erin Pattinson investigated how conserved leaf patterning genes function within the lemma. And her work focused on the gene drooping leaf, which was originally identified as a leaf gene, with loss of function preventing the development of the leaf midvein, resulting in this drooping leaf phenotype seen here in rice. But in rice mutants, loss of function of drooping leaf also results in the inhibition of awn development. And Erin expanded on this work by knocking out drooping leaf in Brachypodium and blackgrass, two species where the evolutions of awns is independent and they have different awn morphologies. So Brachypodium has straight awns initiating at the tip, whilst blackgrass has a more proximally initiating awn that is twisted and bent. But what we see here is that this same gene is being used over and over again and for awn development. And this kind of shows that when the awn evolves, it is not a novel structural invention, but evolution is repeatedly recruiting leaf blade genes for use in awn development. From this work, we hypothesized that not only are leaf and lemma homologous to each other, but the three morphological domains of the leaf are homologous to the three domains of the lemma. However, unlike the leaf, which is fixed into a strict developmental sequence along the proximal distal axis with the sheath, the ligules and auricles, and then the blade, the lemma has been able to somewhat escape these constraints, allowing the awn module to be repositioned or suppressed. While we see some lemmas exhibiting a morphology consistent with the leaf, such as in barley, where the lemma body is proximal and then the awn initiates at the tip. We also see in blackgrass where the awn initiates more proximally, and then in species such as maize where no awn develops at all. So this developmental flexibility allows the awn module to be repositioned, activated, or suppressed without disrupting the rest of the organ and producing a large amount of morphological diversity. And this flexibility at the organ level translates directly into evolutionary lability across the entire family. So Erin's work showed that while the leaf blade is a fixed constant, present in every single grass species, the awn is transient and has been gained and lost multiple times over evolution, with each gain in this figure being illustrated by a pink circle. Not only do we see gain and loss of awns across grass evolution, but we also see a wide range of lemma morphologies. In the cereals, the straight awns of barley and wheat photosynthesize, contributing to yield. Yet we see the same straight awn morphology appearing independently across the phylogeny, such as in the stiper tribe. Similarly, oats have this twisted genicular morphology, which is hygroscopic, and this allows twisting and untwisting of the awns in response to humidity or water, and this aids in seedling burial. But we also see this morphology of a twisted genicular awn within the stiper, and we see more unique morphologies throughout the grasses, such as the feathery awn of Stipa pinnata or the three-pronged awn of Aristida, both of which have been suggested to aid in wind dispersal. So these specialized forms and that provide a range of functions brings us back to our question: why has the lemma been able to generate such diverse morphologies while leaf morphology has remained so constant? We hypothesize that the leaf is trapped under intense selection. All plants need blades to photosynthesize and survive. However, the lemma has been somewhat freed from these constraints, and it's given it the freedom to play around with the patterning of the modules to alter its morphology. And so this leads to the core question of my research, which asks, does the repositioning, activation, or conserved leaf morphology in the lemma drive morphological diversity? And to test this hypothesis, we required a genetic system where we could observe disruptions to this set patterning of the modules. And so that's why we use barley. There's a lot of genetic resources available and a lot of barley mutants with altered module patterning. So the one that we use in the lab is leafy lemma, historically described due to its leaf-like lemma. So the lemma body is much wider, and then it transitions into a wider awn-like awn at the tip that's significantly wider and looks quite leaf-like. And so here we see both an extension of the lemma body and a change in the identity of the awn, which gives it is a good system for us to look at disruption to the module patterning. So looking at our model of leaf lemma homology to the leafy lemma lines we look in our lab, we still see this structural shift. We see an elongated lemma body and a decreased awn length in comparison to the wild type, which is much more compact, and the transition from lemma body to awn is quite obvious. When we zoom in, we see this even clearly. And while it's been hard to define where the lemma body ends and the awn begins in our leafy lemma mutant, work is ongoing to define this more clearly using genetic markers that define the sheath and seeing if they define the lemma body in a similar way. But at the moment, we're defining the transition from lemma body to awn using our leaf homology. So the sheath wraps around the stem and then the blade is flat. So we use the same approach here. So this is the position with which the lemma body stops wrapping around the itself and then becomes flat. So this is what we are defining at the moment as the transition from lemma body to awn. We then wanted to look at this quantitatively, so we measured awn length, lemma body length, and spike across the main spikelet of the barley, with spikelet 1 being the first developing spikelet. And here we see that the awn to lemma body ratio across the development for both our wild type and our leafy lemma. And leafy lemma, we see a uniform drop in the awn to lemma body ratio. And when we look at what is driving this, it is both a decrease in awn length and an increase in lemma body length, consistent with what we saw in our mature lemmas. Something else interesting is that awn to lemma body ratio decreases over time in the wild type, whereas the leafy lemma ratio appears much more consistent, suggesting that there might be something in leafy lemma that is affecting its developmental progress. We next asked that given our predicted homology between the leaf and lemma, we do our lemma phenotypes translate to the leaf. So here again, we measured the same structures, so blade length, sheath length, and leaf position, with leaf 1 being the earliest developing leaf. When we compare these leaves back to our lemmas, we see quite the difference with these phenotypes not being consistent. We see an increase. We see a decrease in sheath length in our leafy lemma mutants, while there is no effect on blade length. And there's a significant decrease in the blade to sheath ratio. So here we show that leafy lemma shows a novel leaf phenotype that's not been described before, with previous work categorizing leafy lemma as a purely reproductive mutant. So to summarize, our wild type, our leafy lemma in the leaf has a decreased sheath and no difference in blade, whilst our lemma body is elongated and our awn decreased. And what this tells us is that whatever's driving these developmental differences and morphological differences in leafy lemma, it's context specific and different in the leaf and the lemma. But we are most interested in what's going on in the lemma. And so to understand the developmental mechanisms underlying this phenotype, we've next looked closely at what is happening in early lemma development. So to look at what is causing our mature lemma phenotype, we looked very early on using scanning electron microscopy. And our goal was to identify when the developmental trajectories between wild type and leafy lemma began to diverge. So when we look at early inflorescence development, here the lemma is false colored in the orange. We find that early lemma primordia follow the same developmental stages and look very similar. But as the stamens begin to differentiate, we first begin to see quite the difference. The leafy lemma inflorescence is much more compressed, and the lemma body less developed compared to wild type. As we begin to see awn initiation in our wild type, indicated by this invagination of the lemma, we do see that the awn in leafy lemma is initiating at a similar stage, but the lemma body is much more reduced. And as we see the awn begin to elongate in our wild type, we don't see this in leafy lemmas, which indicates a developmental delay. And we see this as the awn begins to shoot up in wild type and elongate quite rapidly. There's some beginnings of elongation in leafy lemma, but not to the same extent. And this is consistent with what we see in our mature phenotype, so a reduction in awn length in our leafy lemma. From this, we then wanted to ask what are the genetic mechanisms that contribute to the leafy lemma phenotype. And so we carried out bulk RNA-seq analysis on 3 developmental stages. So an early stage where the developmental trajectories had not diverged yet, suggests which is where pre-patterning mechanisms might be playing a role. A mid stage where the awns begin to initiate, and a late stage where we see this elongation in our wild type, but not in our mutant. So our first step in analyzing our RNA-seq data wasn't to just look at the mutant, but to define what normal developmental trajectories in these stages are in the wild type. And this was because we hypothesized that our leafy lemma would be a developmental mutant, and so any genes involved would be differentially regulated in our mutant compared to our wild type. So first we needed to get an idea of what genes are changing in our wild type over these 3 developmental stages. And so we defined any developmental gene as one that's changed over these stages. So either an increase across the 3 stages, a decrease, or a peak at any particular stage, such as in the middle. And from this analysis, we identified 653 wild-type developmental genes where the expression changes over these 3 developmental time points. So we see some down-regulated and some up-regulated, and then some with peaks in the middle stage. We then expected that any gene that was involved in the leafy lemma phenotype would have a different expression pattern to these wild-type developmental genes, and this shows that 139 genes had a different expression pattern in leafy lemma compared to wild-type. And so we took these genes forward for Go enrichment analysis, and this revealed that cytokinin, lipid transport, and response to cold were enriched. So these are the avenues we are currently exploring, and the questions we are asking now is, what is the biology that is going on here, and what are the mechanisms biologically that are driving these changes in the leafy lemma morphology? And currently we are focusing our efforts on both response to cytokinin and response to cold, but I will just be talking about the response to cold today. So why did we go along this avenue? So we went back to the underlying biology. It is well documented in plants like Arabidopsis that an increase in ambient temperature triggers a dramatic elongation of the petiole, as we can see here. And there are reports of this same elongation response in the grasses as well. So this graph shows that an increase in temperature increases the leaf elongation rate. And because our leafy lemma mutant exhibits the clear proximal distal patterning phenotype and an elongation of the lemma body, we hypothesized that this altered morphology could be driven by disruption to the plant's temperature sensing machinery, which could affect its growth and patterning. So to test this hypothesis in a preliminary investigation, we compared the development of and the lengths of leaves of leafy lemma and wild type grown at 21 degrees or a warmer 29 degrees. And what we see here is that in the wild type, there's no change in blade length in response to temperature, but we do see this consistent sheath length elongation in response to an increase in temperature. And what we see in our leafy lemma is that there is no such response, and there is no significant difference in sheath length between the 21 and 29 degrees treatments. So this suggests that leafy lemma might be insensitive to warm temperature, which results in a disruption to the patterning. And this links back to our transcriptomic data, suggesting that the leafy lemma locus could be involved in patterning via temperature sensing. And when these mechanisms are disrupted in leafy lemma, it results in the different patterning that we see in our lemma. So to summarize the work, we've shown that lemma and leaf are developmentally uncoupled with each other, and leaf and lemma phenotypes in mutants might not follow the exact same morphology. We've reported that leafy lemma shows a distinct leaf phenotype opposite to the lemma that has not been described before. And the fact that the same mutation drives opposite structural outcomes, both an expansion of the lemma body but reduction of the leaf sheath, emphasizes how context-dependent these shared modules are. Our RNA sequencing and temperature experiments have suggested that this might be involved with disruption to temperature-mediated growth regulation. And our immediate future work will prioritize investigating cytokinin's role in this, as well as trying to map the leafy lemma mutant to understand what both the biological and genetic mechanisms that cause this change in morphology. And this will provide deeper insights into how genetic disruptions of module patterning drives morphological innovation. So I'd just like to thank the Bartlett lab, especially Erin Patterson, as she did most of the foundational work on this project, and our collaborators, Anis Richardson and Edinburgh, and Dana McGregor at Romsted. So thanks.
Thanks, Katie, for the interesting talk. From the audience, if you have any questions, you can put in the chat, or you can raise your hand. We will unmute you so you can ask your own question. Maybe I can start a question. So you talk about the ratio of the lemma versus on, or the blade versus sheath. I just wonder if at the cellular level, is the difference is due to cell division or elongation? And have you looked at that?
We tried to look at it just from the SEMs, but it was really hard to get an idea of where the Orm boundary would begin, especially in the early developmental stages. So we're trying new approaches now to get higher resolution and to get an idea of what's causing it, because it must be either whether it's the cells that aren't elongating as much, or there's just less cells. So yeah, that's definitely a thing to look into.
So there is a question. I'll see if I can unmute.
Can you hear me?
Oh yeah, we can hear you. Yeah.
Hello, I'm Alessandro. Thanks, Katie, for your nice talk. My question I wrote in the chat. Is there any effect on temperature also on the length of the leafy lemma, the modified own, or is you have only observed this difference in the leaves?
So when we were doing the work on this, we were restricted on space, so we had to grow them on plates very early on. But I'm currently planning an experiment to grow the plants to full height to see an effect on whether it affects the sensing then. So yeah, I've done separate work to this for a different project, looking at the temperature effects on Brachypodium grown in low and high temperatures, and the Orm and lemma body patterning does change. So it'll be interesting to see whether leafy lemma follows that, and barley in general.
Thank you.
There's a question from Haslop Harrison. Do you want to ask your own question?
Yes. So I've just got the obvious practical question about the awns as to why barley largely has awns, whereas wheat largely doesn't, except for the enormously expensive British variety Skyfall. How is your understanding of the genes involved likely to affect what we see coming through the breeding programs?
I'm not too up to date with the effects on yield. I know from talking to people that it seems the trade-off might be drought resistance with awns versus yield requirements. There's more, or potentially, I think I've seen that it might cause disease or of something if awns are present. But yeah, I'm not too sure how it impacts the breeders too much.
Do you have enough seed of any of these that you could run plot trials 3x1 or something like that?
Not plot trials, but I could just look at the seed and see even if there is a yield impact on this. Because I think, having such a bigger lemma, a wider awn, it might, anecdotally, I've seen that the grains are thinner. So whether that affects it too much and doesn't actually have a yield benefit, we'd have to see.
Yes, because I know there were all the spectacular projects in the 1980s at Plant Breeding Institute looking at the, I suppose, synthetic efficiency from awned species and non-awned, suggesting a lot of the photosynthates came from the awns, but none of that's not the sort of experiment that's done anymore. But your genetic approach is very likely to be able to address some of the effects of the morphology changes. Thank you.
There's a question from Sarah. Sarah, do you want to ask your own question?
Sure. I hope you can hear me. I can't sort out my camera. So I've got a couple questions. The first is, are you going to examine kind of temperature sensitivity to these leaf parameters in different barleys that are adapted to colder or warmer regions to see if they also respond? Or is that something you're interested in?
I haven't thought about doing that. I think we've mainly it was just an idea to see if even response to temperature was involved from what came out of the RNA-seq. But I think it's a good idea, and also just to see if the awns are different in those species as well.
100%. I mean, yeah, it's good to like see what you have, but then I think it could be wildly interesting to kind of start to explore more. And then obviously look to see if some of the genes that you're picking up in your RNA-seq are, divergent, if you've not seen the response or you are, etc. The other one I have was more about kind of your work on diversity. And do you have any plans to look in other awned grasses for forward genetic approaches, and why or why not?
So the other side of the project that was a bit less barley, so I didn't talk about too much here, is looking at blackgrass. So on this, it would be like this type of morphology. And we're kind of unsure at the moment whether all of the lemma would be a lemma or given positioning, and that's how you tend to define homology, is if anything above this might be what the ligule tissues are. So we've been working to try and generate knockout lines in blackgrass to see if that affects the patterning, and then also just to do in situs in blackgrass and other species with different initiation points to see if the traditional sheaf patterning genes are different in those species.
And I don't know enough about blackgrass to know if it's diploid or genetically tractable or.
You can do VIGs in it, and we've been working with the collaborator at Rothamsted to get it. But I think another project in the lab coming up is trying to get it stable transforms, and that would make things much quicker and easy. Well, slightly quicker and easier.
I guess the last, if there's time, more just a comment. So we're in my own group, we're looking at developing some resources to unpick cytokinin signaling. So please get in touch, and I'd be happy to discuss more.
Amazing, thank you.
All right, cheerio. Thanks for the great talk.
There's another question from Jennifer Smith, and she asks if you know the gene or candidate genes that may cause this leafy lemma phenotype.
So we're working with other groups. So groups in Germany and Japan are a bit further ahead, and it's been narrowed down to chromosome 2H, but there might be other factors involved. It's very complicated at the moment, but we're hoping to get an idea soon, and using the resources that the other groups have to get an idea. But I think it's quite complicated, that we're more interested in just what the mechanisms might be, and if we find out what the gene is, that'd be amazing.
Yeah. There's another question from Luke. And Luke, do you want to unmute yourself and ask yourself?
Can you hear me?
Yeah.
Okay. Thanks for the talk. It's not my area, but I was interested on the phenotype you get with the leafy lemma mutant in barley, do you get a midrib in the leafy awn section?
Not one that I have specifically noticed. It's around this, the veining pattern similar to just the normal barley awn. It's just wider, and there might be a few more, because I think the awn in barley has 3 veins, and when we've defined the transition from awn to lemma body to awn, that it has 3 at that point. So that's also kind of what we've been going on, but I've not specifically noticed if there's a mid vein.
No, I'm just from your introduction with the drooping leaf mutant in rice not having awns. It just seems kind of cool. The other thing from this slide you've got up, you've sort of shown an analogy between the ligules, auricles, and the boundary section in your lemma awn. What's the evidence for that? I'm just thinking, for sort of ligulous mutants don't seem to show an obvious grain phenotype or inflorescence phenotype.
Yeah, so this is like the other work I've been working on, is to test this. So we are testing this at the moment. We've got ligulous mutants in Brachopodium, but we've only got ligulus 1, which is quite downstream of it, because that just produces a ligule, whereas ligulus 2 in maize is what does the patterning. So we think maybe if any of them were to be involved, it's likely to be sent quite upstream, involved in patterning, as opposed to producing the tissue. We've seen maybe slight phenotypes in Brachopodium, where there's some tissue at a boundary, but it needs a lot of careful phenotyping, and we're at the moment just checking and to see if these genes are even expressed in the lemma, which will help quite a lot.
Thanks. That's interesting. Thank you.
Can I ask a follow-up question? So talking about the lemma on junction, because in barley it looks like the on looks very similar to the lemma. It's just very narrow. And then in some other species, I think in those wild oat, the on, they look so different. I just wonder if there's any special cellular structures at that junction in different grasses. And did you?
I know people have looked in barley to and have done morphographics because they hypothesized that at the boundary the division rates would be different. Stuff that's happened there hasn't really been able to find anything that clear. I don't think anyone's looked at species with more distinct awns, which is probably, if you're going to see anything specific to define the boundary at the cellular level, is where you would be able to see it. So maybe that'd be a good way to use blackgrass to have a look there.
And what do you think the role of that drooping leaf? Because in the leaf, there seems to be more specific to the midvein. But then it just completely suppressed the on in lemma. So do you think the function of this drooping leaf is conserved, is just function in different places, or it's actually do different things?
I think it's conserved, but it also has other functions as well in the plant. So most in the Brachopodium drooping leaf mutant we have, it's really affects the female floral organs. So there's other, it is effect, it has a role there as well. I think as it's not just the midvein that makes the barley or the awn, there are 3 veins. It can't just be that if a midvein, if you have no midvein, you have no awn, and they're very homologous to each other. So it must be maybe downstream factors or something that are affecting its function in the different locations.
Yeah. Thank you, Katie, for the very interesting talk, and thanks for everyone for joining and the questions. If there are no other questions, we will close today's session. Thank you, everyone.
Thank you.